Megha, you can start now. Dr. Anil is a co-host.
Okay, sir. Good afternoon to one and all present for today's session. It's a privilege to arrange the sessions for Sunday and help out all the research scholars in different arenas and make them aware of what are the opportunities and what are the applications that we can use for making research and research related aspects in a very easy way. So today, yet another Sunday session we are going to have on bioremediation of textile dyes by microorganisms. So first of all, I would like to welcome our today's guest, Dr. Narsinghe sir. I welcome you, sir.
Thank you.
I would also like to welcome our very enthusiastic and very important person, Dr. Arvind Deshmukh sir, for helping and supporting for arranging all the sessions throughout the Sundays without any problem. I welcome you, sir. I would also like to welcome all the participants and the members for today's session. So to start the session before, I would like to introduce our today's guest. Dr. Narsinghe sir has completed his academics from Yogeshwari Vidyalaya, Ambezogai. He has completed his PhD from Dr. BAM University, Aurangabad. Sir at present is serving in Yogeshwari Mahavidyalaya as the head of the Department of Microbiology. To his credentials, sir has attended multiple national and international workshops, conferences, seminars, as well as he has also presented the oral and poster presentations. Sir is very active and has almost 20 research papers published throughout in national and international as well as peer-reviewed journals, and sir is an author for 3 books. So today it's a privilege to have Dr. Narsinghe sir for the session, and I would like to request the participants to please mute yourself and be attentive for the entire session. Over to you, sir.
Thank you, madam.
Sir.
Can you listen to me?
Yes, sir. Yes, sir. You're audible.
Yes, yes. Okay. Shall I start?
Yes, sir. You can start. You can even share your screen as you are the co-host.
Yes. Yes.
Sure.
First of all, I'm very much thankful to the Microbiologist Society of India and Dr. A.M. Deshmukh sir, President Microbiologist Society of India, for giving me this opportunity to speak in front of you. Sir has motivated a lot of budding microbiologists, and sir is the inspiration for all of us. Dr. Deshmukh sir always tries to inspire us. He spread the subject into the different regions, and we are here, and we are, whatever we are doing, it is there is a major contribution of the Deshmukh sir. So I'm very much thankful to the Deshmukh sir for giving this opportunity. I'm also thankful to Megha madam for giving a very nice introduction and for giving this opportunity. So I am here to speak regarding the bioremediation of textile dyes by using the microorganisms. So my topic of the research is the bioremediation of textile dyes from the can you see the screen?
So if you're presenting the PPT, it's not visible.
Is it now visible?
Yes, sir.
Okay.
Yes,
so
it's
my
visible.
topic of research it is bioremediation of textile dyes by using microorganisms. As we know that the textile industries, they are the larger consumers of the water, and these textile industries, they earn a lot of money, and for these, the textile industries, they use a large amount of the dyes that will include the azo dyes. As we know that a dye is a colored substance.
Sir, you have muted your mic. Can you please unmute?
Can you listen to me?
Yes, sir. Now you're audible.
Yes. So dye is a colored substance, and that has affinity for the different substrate to which it is applied. There are many variety of the dyes that will include the azo dyes and many other dyes. These are the synthetic dyes. These are the xenobiotic compounds. These are the recalcitrant compounds, and these are found to be resistant to the degradation. So there are wide variety of uses of these dyes. These dyes are widely used in the textile industry. They are used in the food industry. They are used in the cosmetic industry. They are used in the pharmaceutical industry, paper industry, and in this way, there is a wide use of these variety of dyes. And among all these classes of the dyes, azo dyes is one of the most versatile class of the dyes, and it is one of the most commonly and widely used dyes. So textile industries, they use a large amount of these dyes. And these dyes, they are the aromatic compounds with one or more azo groups, and that will constitute the largest class of the synthetic dyes which are used in the various industries, and that will have the commercial application. As I told, these azo dyes, they are recalcitrant compounds, they are xenobiotic compounds, and when they are discharged into the environment, so they are found to be the resistant to the biodegradation process. And these dyes, they are also found to be very much toxic. As I told, these dyes are widely used in the variety of industries, and majority of these dyes, they do not bind, and that is into the environment through the wastewater which is released from the industries. So that dye, it is getting discharged into the environment, and it is affecting the environment. It is causing the pollution that will causing the hazardous effects to the various ecosystems, to the plants, to the human beings, to the animals. And in this way, these dyes are found to be very much, they are found to be very much dangerous and toxic. The textile effluent, which is released from the textile industry, it is a complex mixture of these variety of polluting substance. So the textile effluent or the textile wastewater, which is released from this industry and which is being discharged into the environment, into the water reservoir, into the water bodies. So this textile effluent or wastewater, it is containing a mixture of variety of polluting substances. So which polluting substances are present in this textile effluent? So organochlorine-based pesticides, heavy metals, variety of dyes, and mostly the azo dyes. These are one of the main component of these textile effluent. It has been reported that many of these azo dyes and the metabolites which are formed from the degradation of these azo dyes, they are found to be toxic, they are carcinogenic, and they are also mutagenic in the nature. That may also lead to the formation of tumors, that may also cause the cancer, that may also cause the allergies. And whenever this dye is released into the environment, means many of the, there are a lot of legislations, government legislations on the release of this textile industry wastewater into the environment. But these legislations and these protocols, they are not being followed by the industry. So a huge amount of this textile effluent or wastewater is being discharged every day into the water bodies, into the water resources without any treatment. And there is a ban on many of such azo dyes. These are banned from the different parts of the countries. There are environmental legislation on the use of these azo dyes, and even then these dyes are used in a large quantity, and they are released into the environmental resources or environmental water bodies and variety of the resources. And therefore, it is very essential that the wastewater or the effluent, it should be treated properly before being discharged into the water bodies. So before being discharged into the water bodies, that wastewater or the effluent which is being released from the textile industry, it has to be treated properly, and then only it has to be discharged into the water bodies, or it has to be discharged into the water resources. When this large amount of the textile water it is released into the water bodies, so that will affect the ecosystem. So when these large amount of the wastewater it is released, so that will be toxic to that particular ecosystem. So as we know that as these are discharged into the water bodies, so that will affect the aquatic ecosystem, that will affect the aquatic life. So as the sunlight, it will not penetrate into the seawater or into that particular water reservoir, and therefore that will also affect the process of photosynthesis. When it is discharged into these water bodies, so aquatic ecosystem, it is also getting affected. The aquatic flora, fauna, fishes, aquatic animals, they get affected. And this dye, it is getting accumulated into the tissues of these particular aquatic animals. So as it is getting bioaccumulated into the tissues of that particular aquatic animals or the fishes, so that will indirectly or that will directly reach to the human beings. So we eat that particular fishes, and that fishes, it is having the residues of these toxic compounds, these pesticides, these dyes, and which can lead to the hazardous effect, which can lead to the variety of cancers, which can lead to the allergies. It has been reported that the workers which are working in the textile industry, they suffer from the human bladder cancer, and that is also one of the major problems. So variety of hazardous effects, harmful effects, they are found to be caused by these dyes. So dyes are mostly the azo dyes, they are toxic, and their metabolites, they are also found to be toxic. When these dyes through the water, through the aquatic animal, through the fishes, when they reach to the human body, so that dyes, they are further degraded by various enzymes in our intestine. So due to the further degradation of these enzymes, or due to the further degradation by the microflora which is present in our body, so the dye is getting converted into intermediate metabolic products. So the metabolites which are produced from these azo dyes, they are found to be very toxic, they are found to be very carcinogenic, they are found to be mutagenic. So after the degradation of these azo dyes, the aromatic amines, they are formed, and these aromatic amines are found to be very much toxic, they are carcinogenic. So therefore, it is very essential that this textile wastewater or the effluent, it has to be discharged into the water bodies only after proper treatment. So there are variety of physicochemical methods, or there are many conventional methods which are used for the removal of the azo dyes from the effluent, but these methods, they have certain disadvantages. So it is very essential to treat this wastewater before being discharged into the water bodies. Some of the methods which are used for the treatment of these dyes, they are found to be expensive. They are very expensive. And it will generate a large amount of chemical sludge. So when it is treated by different physicochemical method, that will cause a secondary problem of the chemical sludge. So secondary waste, it will be generated. And there is another problem of this chemical sludge, the problem of the sludge disposal into the environment properly. So in this way, this various physicochemical methods, they are found to be not that much satisfactory. Again, there is also the disposal problem of these secondary sludge. So that is to proper place. So that will also limit the use of these methods. And these methods, they are found to be very much costly. So and as I told that these dyes, they have been linked to the human bladder cancer. So these dye stuff or these azodyes, they are toxic, they are hazardous. So they are responsible to cause the human bladder cancer. They are responsible to cause the splenic sarcomas. They can also cause the hepatocarcinomas. It will also cause the chromosomal aberrations in the mammalian cells that may also induce the liver nodules in the experimental animals. So in this way, these dyes are found to be very much toxic, and it has to be disposed of into the environment very properly. So these dyes, they are also having the environmental concern. There is a great environmental concern about the fate of these dyes. As the industrial effluent, which is containing these variety of the dyes, particularly the azodyes, so that will reduce the light penetration into the water bodies. So the water ecosystem, it get affected. The photosynthetic activities, the aquatic flora, the aquatic fauna get affected. So in this way, the dyes they are, or the wastewater which is released, it is having the environmental concern, a great environmental concern. So we have to think of the proper method, proper treatment method for the treatment of this particular wastewater, textile wastewater, which is containing the azodyes. These dye-containing effluents that will also increases the biochemical oxygen demand of the contaminated water. And there are variety of environmental legislations on the release of this textile effluent. As I told, the government legislation, it is becoming increasing, it is becoming more stringent, and especially in the more developed countries. So there are government legislation regarding the proper disposal of this wastewater, regarding the proper removal of these dyes from the industrial effluent, and there are some examples, such as the environmental policy in the United Kingdom. Since September 1997, they have stated that zero synthetic chemicals that should be released, that should be discharged into the marine environment. So effluent which is discharged, so that will also have certain regulations in Germany. And these regulations in the Germany, they are among the most stringent in the world. So as compared to the legislations in different countries, the legislations in the India, they are also there, but that is not effectively followed. So there are government legislations, but that should be made more stringent, that should be strictly followed, and that is for the conservation of the environment, that is for the health of the animal, for the health of the ecosystem, for the health of the plant, and for protecting ourselves from these toxic effects of these particular dyes. So as I told, that wastewater which is being discharged into the environment, that should be properly treated. As I told, there are variety of physico-chemical method, but that will have some problem. These physico methods, they are very costly, and there is a need of an alternative method for the proper treatment of this wastewater. So which is that alternative method? So bioremediation of textile dye by microorganism, it is one of the environmental friendly, it is one of the eco-friendly approach for the treatment of textile wastewater and the dyes. So bioremediation, the concept, most of us know the concept of the bioremediation, that we use this living organism, we use microorganism for the detoxification of the waste which is being released into the environment. So that waste or toxic pollutants which are released into the environment, so that can be degraded by the use of microorganism. that can be detoxified by the use of microorganisms. As all of we know that the nature, it has the ability to clean itself. So the cleanup of the environment, that ability, it is possessed itself by the nature. And there are a lot of microorganisms. There are potent variety of the microorganisms which have the potential, which have the industrial application for the remediation of these textile dyes properly, very effectively. And the general approach of this bioremediation concept, it is what we are doing. We are going to improve the natural degradation capacity of these native microorganisms. So these microorganisms, they have the potential to degrade the textile dyes and the effluent, and their ability to degrade it can be improved even by the genetic engineering, or we can make the genetically engineered strains which can have the more ability to degrade the toxic pollutants which are discharged into the environment. So this bioremediation, as I told, it is one of the eco-friendly method. It is one of the cost competitive alternative to the chemical decomposition process. This bioremediation, it is a cheaper method. It is one of the environmental friendly approach or the alternative for the removal of the textile dyes from the textile water, textile wastewater. And there are huge number of bacteria present in the nature which have the ability or which can be used as invaluable tools in the treatment of textile effluent. So there are variety of microorganisms which we can use for the treatment of the textile dyes and for the treatment of the textile wastewater. And we have to exploit these microorganisms. We have to use these microorganisms for treatment of this textile water. There are many microorganisms which have the dye degradation potential. So there are variety of bacteria. So that will include Aeromonas hydrophila, Bacillus benzinovorans, Bacillus cereus, Bacillus licheniformis, Bacillus satilis, Citrobacter, Escherichia coli. We can also genetically make genetically engineered strains which have the ability to produce the enzyme and which can degrade the dyes, or Proteus mirabilis, and many other variety of the strains of the microorganism. They can be used as effective strains. They can be used as efficient microorganisms and which have the potential to efficiently remove the dyes from the textile effluent, which have the potential in the bioremediation of the textile dyes. And we can use these microorganisms very effectively. The topic of my research, it was the bioremediation of textile dyes by using the microorganism. And for this, I have selected the bacterial strain. I have isolated the bacterial strain from the nature, from the dye contaminated site. And the objective of my studies were isolation and screening of the dye degrading bacteria from the dye contaminated soil. So I have isolated, I have screened the bacteria from the site, from the soil which is contaminated with the textile effluent. Then I have performed the identification of these dye degrading bacterial cultures. Whenever we are going to isolate any kind of bacteria which have the potential of bioremediation, so or potential of bioremediation of toxic pollutants, so we need to identify that bacteria. So our main purpose, it is to isolate and to screen the efficient strain of the microorganism. So that will have the more potential of the dye degradation. And then we have to identify these bacteria. So these bacteria, they were identified by the morphological method, cultural method, by the biochemical test. And I have also done the 16S rRNA sequencing by which I have identified that bacteria. Then we have the efficient degradation of the dyes under different conditions. So we have studied the effect of the dye decolorization, the effect of bacteria on the dye decolorization, and we have studied it under different environmental conditions. We have studied the effect of these bacteria under different environmental conditions. and I will tell you. So for this purpose, I have visited to the textile industries located in the Ichalkaranji, Maharashtra, India, and I have selected certain azo dyes. I have selected certain reactive dyes such as the reactive orange, reactive yellow, and the culture medium which I used for my study, it was the mineral salt medium, which was having the pH of 7.2. Then firstly, my task it was to isolate or the enrichment and isolation of these dye-degrading bacteria. So the soil samples which I needed, I have collected the soil sample from the contaminated soil. So the soil from the contaminated soil from the Ichal Karanji I have taken, and that soil, it was used for the isolation, screening of the dye decolorizing bacteria. And I have enriched that bacteria, or I have isolated that bacteria by the enrichment culture technique, by the use of mineral salt medium. And that mineral salt medium, it was amended with the textile dyes. Means whenever you are preparing that media, so even in your laboratory, many students, they can perform this experiment. You can use the nutrient broth. You can add the dyes which are used in your laboratory. You can add crystal violet and many other dyes, and you can inoculate the culture of bacteria, and you can study the effect of that bacteria on the dye decolorization ability. So I have selected the reactive dyes such as reactive blue 59, reactive golden yellow 84, and reactive orange 96. So I have used 50 milligram per liter of that dye. It was then added into the 100 ml of the culture medium, MSM medium, which was taken in the 250 ml Erlenmeyer flask. And then I have inoculated the 10 ml of the 10 gram of the soil sample. The soil sample, or we can make suspension of that soil sample, and we can inoculate the 10 ml suspension or 10% into that medium, which is amended with textile dyes. And that dye containing medium was inoculated with the bacterial culture or the soil sample, and then it is incubated in the incubator at 37 degrees Celsius. And the samples or the media which showed decolorization in that liquid media, they were repeatedly further tested.
Sorry for the inconvenience. The speaker by mistake has left the Zoom meeting. Please be there. I'll try to put him back into the session. Sorry for the inconvenience, members.
Can you listen to me?
Yes,
Hello?
sir. Yes, sir. You're audible.
Okay, there was internet disconnected. Sorry for the interruption. May I continue, ma'am?
Yes, sir. Please continue.
Sure.
छोटी रे बयान कथा छोटी आणि काय पाहिजे का काही नको.
I have isolated the efficient strain of the microorganism from the dye contaminated soil, and then I have further tested consistent bacteria are showing the consistent growth, and those bacteria which are showing the consistent decolorization in every successive transfer. I have used that culture, and an aliquot of 0.1 ml of that sample Then I have taken allocate of the sample which showed the consistent growth and the decolorization in that particular MS medium, and that 0.1 ml it is then inoculated onto the MSM agar plates, which is containing 50 milligram per liter of that particular dyes. And then after incubation, the colonies which are showing the zones of decolorization, that colonies were selected. Again, those colonies which are showing the large zone or the diameter of the zone it is more, that colonies they have selected. Those bacteria which have the ability of consistent growth in the dye containing medium, which have the ability of maximum decolorization, that were selected for the further studies. Whenever we are going for such isolation of the bacteria, so our purpose it is to isolate, to get the efficient strain of the bacteria. And we have isolated a large number, nearly 100 strains they have been isolated. And from among that, I have selected only 2 strains, which have the ability of maximum dye decolorization and degradation. I have also gone for the screening of the dye degrading bacterial cultures. And from among that, I have selected the most potent dye degrading bacterial culture. I have also studied the effect of various environmental factors such as pH, temperature, carbon sources, nitrogen sources, variety of inducers. And I have tested the effect of these environmental factors on the dye degradation ability of the bacteria. So these factors were tested, and I have done the decolorization assay. So initially in the laboratory, I have performed the decolorization assay by using the UV visible spectrophotometer. So the supernatant, which is obtained after the centrifugation, so that supernatant, it was then withdrawn at the different time period. So the medium which was inoculated with the dye and which was inoculated with the bacterial culture, after incubation, we have centrifuged it, we have obtained the supernatant. Supernatant, it was withdrawn at the different time intervals, and we have measured the maximum absorbance at the lambda max of 600 nanometer for these particular dyes. Each dye that will have that particular lambda max, and we have taken the readings, and the percentage decolorization, it was then calculated by the formula from the difference between the initial and final absorbance. So initial absorbance, it was also taken, and then it was divided from the final absorbance, and all the experiment they were performed in the triplicate. The decolorization, it is expressed in the terms of percentage decolorization, and that percentage of the decolorization, it was calculated by using the formula. You can see the formula, that is percentage of decolorization is equal to initial absorbance minus final absorbance upon initial absorbance into 100. And by using the formula, we can get the percentage decolorization of that particular dye. Then we have performed the identification of the bacteria, dye degrading bacteria. At the laboratory scale, we have performed the morphological characteristic, cultural characteristic, biochemical characteristic, and by using these, we have tentatively identified that bacteria. And it was confirmed by the confirmation, it was made by using the 16S rRNA sequencing analysis. This 16S rRNA sequencing analysis I have performed at the Reddy's lab in the Pune, and there I have performed this particular sequencing analysis. And then we have obtained the results from among all the bacteria which we have isolated. Only 2 most effective strains they were selected for further studies. And then this is the flask, or the image which is showing the flask, which is showing the decolorization by the bacterial cultures. Then we have studied the effect of the different concentration of the dyes onto the dye degradation ability of the bacteria. So I have identified the bacteria, and the bacteria were found to be the Bacillus licheniformis and Rhizobium. And I have studied the effect of...
Anil sir, you have to move this slide, please.
Can you see?
Yes, sir. Oh, actually, only one slide we can see.
Okay. You can't see the slide now. Table, can you
Sir,
see the
the
table?
slide is visible, but it's not moving according to what you're trying to explain.
Okay, okay, okay.
And if
Now-
possible, please do
Can you see now?
slide view. Yes, sir. Now it's visible.
Yes, I have studied the effect of different concentration of the dyes on the dye degradation ability of the bacteria. And I have used the concentration from 50 milligram per liter. So I have increased it up to 350 milligram per liter. In case of the Bacillus licheniformis, which was found to be the most potent strain for dye degradation, and that strain have the ability to degrade 150 milligram per liter of the dye.
It seems sir has some technical problem. Please hold. Sorry for the inconvenience. I guess sir has some network related issues. Please stay online, all of you. Sorry for the inconvenience.
Okay, can you see, ma'am?
Yes, sir. Yes, sir.
Sorry,
Slides
there
are
is
not visible.
yes, there is net problem. So can you see the slide?
Yes, sir.
So I found that both the bacteria Bacillus licheniformis and Rhizobium that was the having ability of the efficient degradation of the dye, and Bacillus licheniformis it was found to be the more potent. So as we go on increasing the concentration of the dye, so there was increase in the percentage degradation of the particular dye, but it was up to certain extent. After 150 milligram, we found that there was decrease in the percentage degradation, and 150 milligram it was found to be used by the Bacillus licheniformis. 100 milligram per liter it was used by the Rhizobium species. Then we have studied the effect of pH on the growth of these bacteria. So we have studied the effect of pH on the growth and dye decolorization ability of the bacteria. We found that the pH from 1 to 10. Wait. So we have selected the pH from 1 to 10. And I found that the pH 7, it was found to be optimum for the degradation, efficient degradation of the dye by the both strength. Since the optimum pH, it was found to be pH 7. But even if then these bacteria, they have the ability to degrade that particular azo dye over a range of pH. So that was the advantage. Whenever you are going for isolation of any bacteria or any strain, so that should be grown or that should have that potential at a wide range of conditions. So then it is found to be the efficient. And in my case, in my study, in my work, I found that both of the strains, they were found to be showing efficient degradation of dye at the wide range of pH, wide range of temperature, at wide range of concentration of the dye. Then I studied the effect of different temperature on the dye degradation ability of the bacteria. So I have selected the temperature from 30 to 50 degrees Celsius, and I found that the 30 degrees Celsius, 37 degrees Celsius temperature, it was found to be the most optimum for the dye degradation by the Bacillus licheniformis. And for the Rhizobium species, 30 degrees Celsius, it was found to be the optimum temperature. So for Bacillus licheniformis, 37 was the optimum for rhizobium species. 30 degrees Celsius was the optimum temperature for efficient degradation of the textile dyes. Again, I found that our isolated strains, they have the efficiency to degrade that particular dyes over a range of wide range of temperature. Means it can even degrade the dye up to the 50 degrees Celsius. Yes, definitely the percentage decolorization, it will be decreased. As we go on increasing the temperature, the percentage decolorization ability, it will be decreased, but it have the potential to degrade the dyes at a wider range of temperature. Then I have also studied the effect of a different concentration of the sugar on the growth and dye degradation ability. So I have studied the different variety of the sugar such as glucose, lactose, maltose, fructose, sucrose, starch, dextrose, sodium acetate, and I have studied their effect on the growth and dye decolorization ability of the bacteria. I found that the Bacillus licheniformis, they use glucose efficiently, and by using glucose, they have shown the maximum percentage decolorization and degradation. Bacillus licheniformis has shown 77.72 degradation by using the glucose, and the rhizobium, it has used the dextrose as a sugar, and it has shown the maximum decolorization. So I have also studied the variety of these carbon sources and its effect on the percentage decolorization ability of the bacteria. So in this way, I have studied the effect of different environmental factors on the growth of the particular growth and the dye degradation affinity. Then in the laboratory, I can visibly seen that there is a dye decolorization. So by seeing the flask, you can see that the color is converted into colorless form. So there was a decolorization, but then I have done the degradation. I have analyzed the degradation by the UV visible spectrophotometer, and then further I have confirmed that dye degradation by GCMS analysis. So whether actually there is the dye degradation or not, to confirm that, I have performed the GCMS analysis. I have performed it at the University of the Pune, and I have sent the samples to the University of Pune, and I have analyzed the GCMS to analyze the dye degradation ability of the bacteria. And after the GCMS analysis, it was found that a variety of metabolites were formed after the degradation of that dyes. So when we add the dye into the medium, so whether the dye is actually getting degraded or not, and whatever the metabolites which were formed, whether these metabolites they are formed, whether these metabolites they are toxic or non-toxic or less toxic, that we have studied and we have analyzed by the GCMS analysis, and I found that the dye is completely degraded into the variety of metabolites, and that their toxicity, it was found to be very much reduced. And in this is the figure which is showing the GCMS chromatogram of the metabolites, which is formed after the degradation of the reactive dye, reactive orange 96. In this way, I have analyzed the GCMS for each dye, and then I have analyzed the metabolites which were formed after the degradation. Can you see the slide, ma'am? Hello.
Yes, sir. Yes, sir. It's visible.
Okay, so this is the mass spectral profile of N4 amino acid aziridine. This is the mass spectral profile of the 9-oxabicyclo nonane 1,4-diol. And in this way, I have analyzed the degradation of the particular dye. I have confirmed that dye is completely degraded into the different metabolites. Then I have identified the bacteria by using the 16S rRNA sequencing. So at the lab scale, by performing the cultural, morphological, biochemical tests, I have tentatively identified the bacteria, but it was confirmed and it was further identified by the 16S rRNA sequencing analysis. and this is the phylogenetic tree. And our isolated and efficient bacteria, it was found to be showing the close relationship with the Bacillus licheniformis, and it was found that the Bacillus licheniformis, it is the isolated bacteria which have the more potent ability to degrade the textile dye. So the Bacillus licheniformis was found to be the most potent application in the dye degradation or in the removal of the textile dyes from the wastewater. Then I have found that the another strain, it was found to be the Rhizobium. So Rhizobium, it was also efficient dye degrading bacteria, and I also have performed the phylogenetic tree of that Rhizobium, and that was also found to be the efficient strain of the bacteria. So in this way, I found that among the strains, all which I have selected, means from the 100 samples which I have tested, only 2 bacterial strains were found to be efficient. They were efficient in the dye degradation ability. They were having the ability to efficiently degrade the dyes at a variety of conditions, at a wide range of conditions, and that can be used at the industrial scale, that can be used for the industrial application, for the removal of the industrial effluent from the textile industries, from the textile wastewater. So in this way, this is the work which I have performed, and I was got the most efficient strains, and that can be used for the industrial application. And these were the most potent strain which were having the most bioremediation ability. So thank you very much. So whatever the slide which I have prepared, whatever I have studied, whatever the work which I have done, so I tried to present in a short period of time. It is not possible to show or to discuss all the work, but
Yes,
due to the lack of the time, I
Yes,
tried
sir.
to present this in a very less time, and I tried to give my better. I don't know how much the students and you all get it. Thank you very much, Deshmukh sir. I'm very much thankful again to Honorable MSI President Dr. Deshmukh sir, so for giving me this opportunity to present, to give my talk on bioremediation. And sir always motivate us. He is an inspiration for all of us. And I'm able to give this talk only because of him. I am also thankful to the Megha madam for giving this opportunity. Once again, thank you all.
So indeed, it's our pleasure. We'll move on to the question and answer session. Anyone wants to ask any question can unmute yourself and ask the question. Someone was asking.
Yes, sir.
You can please continue.
Yes, please.
Sir, I am Dr. Tanmaya Das from West Bengal.
Yes.
Sir, I have a question. Sir, how did you sterilize dye-containing media? Is it autoclavable, or you have filtered only dye and you have autoclaved media? How did you sterilize the dye-containing media?
Yes. So I have prepared the medium. I have sterilized the medium, and after proper sterilization, I have added that particular dye samples into that particular media aseptically. So aseptically, I have added the dyes, and initially the media, it was sterilized properly, and then I perform my experiment.
I have my question is, you have autoclaved MSM media separately, and or the media or dye both are autoclaved?
No. Yes, you can. We can also both can be autoclaved. So these dyes, they are actually resistant to the high temperature, so their structure will not be damaged.
Yes.
So both can be sterilized. So there is no problem of the issue of the damaging the structure of the dye.
Okay, sir.
Yes.
And
Hello. Yes, ma'am. Continue.
Yeah, so autoclave is the main method for sterilization for media, and so I can use the autoclave for dye containing media also.
Yes, definitely, ma'am. Yes, you can use.
Okay, okay. Actually, some of the paper says the dye should be sterilized by filtration. So I have a query, so I am asking you.
No, I didn't. I have not performed that because in many of the references. So why the azo dye is particularly used in the textile industry? Because that will bind and that will be resistant to degradation and all condition, high temperature. So while processing in the textile industry, high temperature it is actually applied there, used there. So there is no issue that. Yes.
Okay, just like the Congo red, brilliant blue, brilliant green, that may be
Yes,
autoclave.
particularly I'm telling about particularly azo dyes.
Yes, yes, yes. Okay,
Yes,
okay. Thank you,
yes.
sir. So I can use
Thank you.
autoclave
Yes,
for
ma'am.
sterilization.
Yes,
Thank you.
yes, definitely, ma'am. Yes, definitely.
Thank you, sir.
So the
Welcome.
next question is from Prakash sir. What is the mechanism of dye degradation by bacterial cultures?
So the bacteria, they will have the ability to degrade the dye. Many bacteria, they produce the enzyme azoreductase. So the enzyme which are produced by the bacteria such as the azoreductase. So with the help of that enzyme, they can cleave that dye, and that is having that application. Or in case of fungi, such as the white rot fungi, they are the most potent. So these fungi, they are the most potent dye degrading fungi, and they can produce the variety of enzymes such as lacase, or some of the fungi, they can produce the lignin peroxidase enzyme, with the help of which the dye is degraded efficiently.
Okay, sir. The next question is from Amrish sir. He
Yes.
wants to know the composition of the medium used for the isolation of the dye degrading organisms, and also he's interested to know the incubation period for its enrichment.
Yes, in this regarding the composition of that particular dye. So the dye contain variety of minerals. So that will contain the composition of the dye, the medium it contain K2HPO4 1.6 gram, KH2PO4 0.2 gram, ammonium sulfate, magnesium sulfate, ferrous sulfate, sodium chloride, calcium chloride, and one of the most important thing that you can also test the all these minerals or these elements, and then you can add the dye, you can study the effect. So without glucose or without carbon source, you can also study. So that will help whether the bacteria, it will use the dye as the sole source of carbon. That will be helpful for that. But in my experiment, I found that the dye degradation ability, it found to be for the dye degradation ability, there was a necessity of the carbon source. So that's why I have tested, I have added carbon sources such as glucose and many other sources, and then we can study. So we can, without adding the carbon source, you can test. So if it is showing, that is much better. That will show that the diet can be used itself solely as the carbon source. And by adding the carbon source, you can also test it.
Hello, sir. Hello.
Yes.
I have
Yes,
another
sir.
question. Sir,
Yes.
this degradation you are telling dye degradation. So what is the
Yes.
mechanism? Like it is degrade completely, or any secondary metabolites or any end product they are going to release? Is it going to degrade completely so that that will be non-toxic by
Yes,
the
sir.
bacteria?
Means it is not sure to give the 100% surety that the dye which is degraded into the metabolite, it is completely non-toxic. But in my case, I found that the metabolites which were released, they were found to be of the less toxic form. So that I didn't assure or give the surety that it is completely degraded, but toxicity, it is very much reduced. And we have to work further. If to test that, we need to work further, so we can know that definitely.
Sir, in MSM, you are told the composition. Let me
Yes.
have the composition. Later on, you can Means I will communicate to you. But
Yes, sir.
whether this dye is bioavailable or non-bioavailable to the bacteria, this dye during the degradation.
Bioavailable means I'm not getting the question.
This one, sir. This dye, sir. Whatever you use the dye in the media. You have enriched the media with a dye, sir. Hello.
Yes, sir.
You
Yes,
have
please,
used
sir.
the media. No, sir.
Repeat your
This,
question, sir.
dye you have used with the media, sir. Dye. You have used the dye, no? As a dye.
Yeah.
So
Yes,
whether
sir.
it is completely bioavailable to the bacteria or as a sole source it is using or as a it is going to degrade only.
No. In my case, I found that there was a need to add additional carbon source for the complete degradation of the dye. I didn't get any bacteria which have the potential to use dye itself as a source of carbon and it degraded it.
Okay, sir. Okay.
Is it clear, sir? Yes.
Thank you.
Sir,
Thank you.
once more, I want to add it. So, can we use nutrient agar plus dye for growth of the bacteria?
Yes.
Can we use nutrient
Even in
agar?
lab skin, yes, no problem. You can amend that nutrient agar medium with the dyes.
Yes, yes.
And you can also test it. That will also give the results. No problem.
Okay, okay. Thank you.
Yes.
Sheetal Gandhi, ma'am, you can ask your question.
Okay,
Yes.
sir, I have one question. I was doing experiment on dye degradation in methylene blue dyes. I mean triphenylamine dyes. I had used two dyes, crystal violet and methylene blue. What were my observation? In case of methylene blue, I mean dye degradation was above 80%, but not above 90%. Because after 7 days also, I was observing a thin layer of methylene blue in the upper part of the test tube. So I read a lot, but I didn't get any prominent answer. So if you know anything about that what is that layer? I mean, why? Like if you'll shake the test tube, then again there will be a blue color penetrating the entire test tube. So I just wonder if you have any idea why it is so, because it was
You
like
need to confirm
not
it
about.
whether actually in many cases that may be the bioadsorption also. The diet is getting only absorbed to the bacterial biomass. So that is not the complete degradation. You need to further test this whether there is actual degradation by UV visible spectroscopy and then GCMS analysis which I have performed.
Sir,
So you
I
need
had
to
okay. I did FTIR, and I found stretching at C and CN, C double bond N. I mean carbon and amino stretching, CH and NH2. And it means that maybe aromatic amines were formed, but still there was like not 100% degradation. I mean, when you observe, you will find it is above 90, but when you shake the test tube, against there is reoccurrence of color. Reoccurrence of color was, which is what I observed. So I was not sure about why, how and why it is happening.
Yes. Many times you have to further test it. That will show that whether actually it is aromatic amines are formed. So for that only GCMS and other methods are there by which we can know it.
Okay, and one more question, sir. After degradation, I mean, after 6 or 7 days, the culture with dye, I mean, MSM media with dye used to become a little bit sticky. So can I conclude that this may be because of the formation of exopolysaccharides by bacteria under stressful conditions?
That
Yes,
maybe
yes.
I can't.
Yes, you can conclude. According to me, if
Yes,
the
sir.
culture after 7 to 8 days is sticky,
Mhm.
definitely it is producing exopolysaccharide. And
Okay.
that is the most probable reason why it becomes sticky. And second question you have asked, 90%
Yes, sir.
degradation, but not
Yes,
10%.
sir. Yes, sir.
So theoretically, there should be 100% degradation.
Yes,
And
sir.
the flask or test tube should be kept on shaker. If it is
Yes, sir.
kept on a shaker, 100% degradation you will get.
Yes, sir. But actually, what I observed, I mean, I studied both the conditions, static as well as shaking conditions. But in my case, better results were in static conditions as
Okay.
compared to shaking. So
Correct.
maybe there, my bacteria may be facultative anaerobes or
Yes, yes, yes.
anaerobic.
Correct. Maybe facultative. That's why you are getting good results in static. That is very good.
Yes, sir. But in
Okay.
case of methylene blue, reoccurrence of color was observed. So I was a little bit like, why it is happening? I studied a lot, but I didn't got any answer. So like in today's session, I thought let me ask sir as because he has done a very good research in this.
Yeah.
So the next
Okay.
question is from Lakshmi ma'am. When we try to remediate dye with the help of microalgae, we normally let them grow in presence of dyes and light. But there are some dyes that decolorize faster in light, such as malachite green. So how can we justify the role of microalgae here? So she wants to understand the role of microalgae in degrading the dyes faster than the bacterial cultures.
Okay, okay.
So she is asking the reason behind that.
Yes, sorry to say, but my work it is on bacteria. I hope Deshmukh sir may also give a better answer to this question. Sorry for this. I have work on the bacterial cultures.
Yes, sir. Yes, sir. Deshmukh sir, would you like to answer the question? I guess sir is in journey. Lakshmi ma'am will get back to you with the answer
Hello.
for the same.
Hello.
Yes, sir. Yes, sir.
What question you want us to ask me?
Sir, Lakshmi ma'am wants to ask that when we try to remediate dye with the help of microalgae, normally the dye gets degraded faster in presence of light. So what is the reason behind the algae being faster in dye degradation than the bacterial cultures? The reason behind that?
What I can suggest her, she asked the same question, eminent mycologist of India working on microalgae degrading dye. He will be able to answer you perfectly.
Okay, sir. We'll pass. We'll give her the answer.
Correct. Because
Yes.
in Trichy,
Okay.
scientists are working on microalgae.
Okay, sir.
So I will give their number and they will be perfect answering the question.
Sure, sure. Lakshmi ma'am, if possible, please contact Deshmukh sir for your answer. Sir, the next question is, how do you compare activated sludge process and membrane bioreactor system for the dye degradation? Narsinghe sir?
Yes, activated sludge. No, I have
Yes.
not worked on this aspect, so I may
Okay, sir.
not be able to justify it.
Okay, okay.
Yes, but you see, activated sludge process definitely enhance biodegradation of the color, because in many sugar industries, activated sludge is the common process for dye degradation of the industrial waste.
Yes.
So here in the sugar in natural colors here, not the synthetic color, but activated sludge process definitely enhance any aromatic compound and degrade it perfectly. So activated sludge process is a nice in degrading the dye. Okay.
Yes, sir.
Hello.
Thank you, sir.
Yes, sir. Yes, sir. You're audible.
Okay.
Narsinghe sir, the next question is, dye degradation is through bioaccumulation or biosorption?
In my case, there was a complete dye degradation, and which was found to be confirmed by the GCMS, that shows that there was a complete mineralization, means complete degradation of the dye.
Okay. And the next question asked by Amrish sir is, can we have alternative to chemical dyes that can be eco-friendly?
Yes, many natural dyes, or initially also the dyes from the natural plant materials, animals, or they were extracted and they were used. So definitely the natural resources, they are the alternative, best alternative. But there are properties that definitely the chemical dyes, they might be having, they are having the good properties. So that is the disadvantage of that natural dyes which are derived from the natural sources.
Okay, sir. Lakshmi ma'am, can you please unmute yourself?
Yeah, okay. Thank you, ma'am. So that was a wonderful session. And my question was exactly, did you check the influence of light on dye degradation? Because I'm right now doing my research in microalgae. So I grow algae in growth medium, as you said, like for bacteria, we have MS media or nutrient agar, right? So like that for microalgae, we have BG11 medium. And so when I'm growing my algae in the growth medium in presence of dye, we need light. It needs light to grow. So what I say is, in a day or 2 days, the control dyes in which there is no algae, that itself is getting decolorized. So like, do you think, is there any point if I choose such dyes, for example, malachite green?
Can I add something on this?
Yes, yes, ma'am.
Yes.
Uh, Actually, like I'm working at a place where we do oxidation, also oxidation of dyes. So maybe the decolorization may be due to photo oxidation, since you are exposing dyes and algae obviously in front of light. So because of the presence of light, the oxidation is fast. So
Yes.
this
Yeah, but
can
then
be
I
one of
have,
the reasons.
yeah, ma'am, that's right. But then I have read papers on degradation of malachite green using microalgae, and they have used very less concentration, like 10 mg per liter. So I really wonder, because when I'm using low concentration of malachite green, in presence of light itself, it's getting decolorized. So I'm really wondering, like, yeah.
So actually, Lakshmi, what I have also done degradation studies on malachite green along with many other dyes. So what I observed in case of malachite green, when I was just started, apart from light and other things, as soon as you start inoculating the bacteria, at the same moment, decolorization start in case of malachite green. This is what I have also observed.
Exactly, yes.
As soon as at the same moment, just you added the bacteria, you will start observing decolorization by visibly. I mean, it may be in terms of like 1 or 2%. So I was
Yeah.
also surprised. Maybe malachite green is, I think, sulfate containing dye. So it may be.
Sheetal ma'am, sorry to interrupt. We need to ask few other questions. It will be better if both the
Okay.
ladies can contact each other
Yes,
and
yes,
solve the question.
yes,
Sorry
yes,
for the
yes, ma'am.
interruption.
Okay, okay.
Thank you, ma'am. Thank you.
Yeah. Any other questions, please? Okay, so fine. There are a few more questions, but one more question, sir, from Amrish, sir. Like he's asking that I heard biopigments produced by bacteria and fungi are used in textile industry. How far it's true?
Biopigments.
Yes, sir.
Yes,
I did
yes.
not-
What I can tell you, the biopigment is a separate topic. Now, vast application, and it is at a research level. But as one student have told that whether they are eco-friendly, yes, they are eco-friendly. And as again Narsinghe sir has told, the chemical pigments are having better properties as compared with the natural pigment. That's true. But attempts are being made, and whether they are used in industry, as per my knowledge, yes, they are used. But in which industry, you can search on Google and you can find out. On Google, it is easily available. Knowledge is easily available. Okay,
Yes,
thank you.
yes. I guess the questions will
Yes.
not get over.
Now,
So,
now you can conclude.
yes, sir. Yes, sir. So it was a privilege to have Dr. Narsinghe sir for today's session. It was a really knowledgeable session, sir. Thank you for your valuable time as well as your information. And we hope that if any questions or queries raised, it's okay if we share your number to the people so that they get proper guidance with respect to dye degradation, sir.
Yes, definitely, ma'am.
Yes, sir. We are very thankful. So I owe a great thanks to you, Dr. Narsinghe sir, as well as I also thank Dr. Deshmukh sir for unended and very enthusiastic support for all the Sunday sessions. And last but not the least, I would like to thank all the members to be so active and make this session really fruitful by asking the questions and even by participating very actively in the session. Thank you, sir. Thank you, Narsinghe sir. Thank you, Deshmukh sir. Even thanks for all the participants. We can end the session, sir.
Yes, yes.
Yes, sir. Thank you. Thank you, sir.
Thank you, ma'am. Thank you, sir.
Yes, sir. Yes, sir.